What about with this picture? How many of each type of white blood cell are there? It's a little bit more challenging when there's more cells to consider. But this raises the question, is there a way to do this faster? As mentioned before, your eye automatically notices that there are larger white blood cells and smaller white blood cells. Your eye automatically notices that the cytoplasm looks different between the two cells. Your eye automatically notices that the nuclei are different between the two types of cells. And so let's keep track of this. We compare large cells versus small cells, and cytoplasm, whether there's granules or not, or the color of it, and the nucleus. We can combine, or we can transform these characteristics into size, that's large versus small, and then the texture of the cytoplasm and the shape of the nucleus, we can combine into a term called internal complexity. If we made a graph of size versus internal complexity, we could easily plot these four white blood cells on this graph as shown. The neutrophils are larger in size and higher in internal complexity, and the lymphocyte is smaller in size and lower in internal complexity. And since we've already represented what they look like, we don't have to have the actual pictures. We can replace them with a dot. So is there a way to organize the white blood cells from this picture a lot faster? Yes, there is. And we can convert it to a chart that looks like this, where we see 12 neutrophils that are larger in size and have higher internal complexity, and 3 lymphocytes that are lower in size and lower in internal complexity. Now, these terms, size and internal complexity, we can convert to forward scatter and side scatter. These are the terms that are typically used in flow cytometry, but that is what they represent, size and internal complexity. Let's do this for another case. Here, the lymphocytes look pretty similar. They're all small in size and small and have little internal complexity. But this begs the question, what else can we do? If we were looking at this within a surgical pathology world, we could raise the question of where are the B cells and use immunohistochemistry to try to answer this question. Using a blue counterstain and a brown immunohistochemical stain, we could use an antibody against CD19 to identify B cells. And then in another slide, use an antibody against CD3 to identify T cells. Then ask where are the abnormal markers and use CD5, for example. And putting these together, we can make the conclusion that B cells have aberrant CD5. However, is there a faster way to do this? Using individual IHC, we had to compare between CD19, CD3, CD5, back to CD19, comparing slide and going back and forth to figure out which population had which stain. What if we did this within the same slide? Say we used one color for CD19, represented by a yellow triangle, another color for CD3, represented by the blue square, and a third color for CD5, represented by the red triangle, red diamond. We could then make plots or charts of one marker versus the other, say CD19 versus CD3, or CD19 versus CD5, and use some fluidics, lasers, and optics to plot all of these cells on a chart. Here's one way, where we isolated CD19 positive cells or expressing cells compared to the CD3 expressing cells. And we can have another iteration where we look at CD19 versus CD5. With this, Flow cytometry is our digital eye.